Greetings, fellow cosmic voyagers. Tonight, as the universe continues its silent, grand performance, we turn our gaze inward to a microscopic marvel hidden within a substance so fundamental, so ancient, it has cradled life for millennia. We're talking about milk, yes, the humble liquid, but beneath its comforting facade lies a secret architecture, a complex membrane teeming with compounds that whisper promises of profound impact on our very being, from the dawn of our cognitive abilities to the resilience of our bodies. Join me as we unravel the extraordinary tale of the milk fat globule membrane. Settle in, perhaps with a warm drink, and let's journey into the intricate science together. If you find yourself drifting into wonder, remember to like, comment, and share this exploration with others who seek knowledge in the quiet hours. Welcome, curious minds, to another journey into the intricate wonders of the universe, a universe that, as we often find, is as vast within us as it is beyond. Tonight, we delve into a remarkable, yet often overlooked, biological marvel. The milk fat globule membrane, or MFGM. It's a name that might sound purely technical, perhaps even a touch mundane, but I assure you, within its delicate structure lies a profound story of life's ingenuity, a story woven into the very fabric of mammalian existence, from the smallest infant to the oldest elder. Imagine, if you will, the humble drop of milk. For centuries, millennia, even, it has been a symbol of nourishment, sustenance, and the very essence of early life. But beneath its milky surface, within its microscopic architecture, resides a hidden complexity that continues to unravel itself to scientific inquiry. At its core, the MFGM is a delicate, yet incredibly robust envelope. It's composed primarily of lipids and proteins, and its fundamental purpose is to encase the tiny fat globules secreted from the specialized cells responsible for milk production in humans and all other mammals. Think of it as nature's perfectly designed packaging, ensuring the vital fat within is not only protected but also delivered with an entire entourage of bioactive compounds. These compounds are not just passive passengers. They're active agents, a veritable treasure trove of biological sophistication. We're talking about phospholipids, sphingomyelins, gangliosides, glycolipids, glycoproteins, and carbohydrates. Each of these components, with its own intricate molecular dance, plays profoundly important roles within the brain and body. They are the unseen architects of our development, the quiet guardians of our health. For decades, the scientific community has been captivated by this intricate membrane. Over 2,000 scientific publications have explored its nutritional and functional benefits, painting a vivid picture of its widespread influence. Clinical trials, the rigorous crucible of medical evidence, have illuminated its remarkable capabilities. In infants, MFGM supplementation has been shown to support crucial cognitive development, laying the groundwork for future learning and understanding. For adults, it offers a tangible boost, improving memory and even enhancing mood, a testament to its profound connection with our neural pathways. And for our cherished older adults, it has demonstrated an ability to enhance muscle strength, improve balance, and even sharpen agility, providing a vital bridge to sustain mobility and independence. Beyond these direct human trials, preclinical studies, conducted in carefully controlled environments, have allowed us to peer even deeper into MFGM's mechanisms. These investigations have revealed its significant effects on fundamental brain structure and function, on the intricate development of our intestinal systems, and on the robust defenses of our immune systems. The evidence is compelling, spanning the entire spectrum of life, from the fragility of premature infants to the boundless energy of preschool-age children. Dietary supplementation with MFGM, or its key components, has been consistently linked to improvements in cognition and behavior, a healthier gut and oral bacterial composition, a reduction in fever incidence, and a demonstrable decrease in infectious outcomes, including common afflictions like diarrhea and otitis media, the dreaded ear infection. But the story of MFGM doesn't end there. Its influence extends even to the realm of cardiovascular health. Emerging research suggests it plays a vital role in modulating cholesterol and fat uptake within the body. Clinical trials in adult populations have provided promising insights, indicating that MFGM could positively affect markers associated with cardiovascular disease. This includes the lowering of serum cholesterol and triglycerol levels, and even a reduction in blood pressure, a silent guardian working to protect the very engine of our existence. So, how does this remarkable structure come into being? The journey of MFGM is as unique as its benefits. It all begins within the specialized epithelial cells of the lactating mammary gland, cells affectionately known as lactocytes. These microscopic factories are masters of lipid secretion, employing a mechanism distinct from nearly all other non-mammary cells in the body. This distinction is crucial, for it's precisely why MFGM is unique to milk and not found in other non-dairy food products. The process unfolds in multiple, exquisitely choreographed stages. First, fats synthesized within the endoplasmic reticulum, the cellular machinery responsible for lipid production, begin to accumulate. These nascent fat droplets form between the inner and outer phospholipid monolayers of the endoplasmic reticulum membrane. As these droplets grow, the two monolayers gently separate, eventually pinching off to fully encapsulate the droplet within a single phospholipid monolayer. This clever design allows the fat to disperse freely within the watery cytoplasm of the cell. In the next critical stage, these lipid droplets embark on a migration, moving purposefully towards the apical surface of the cell, the outermost boundary. Here, another extraordinary event takes place. The cell's plasma membrane, its outer skin, begins to envelop the migrating fat droplet, slowly but surely extruding it from the cell. As this extrusion occurs, the plasma membrane provides an additional, complete bilayer of phospholipids, effectively encasing the fat droplet in a sophisticated, multi-layered shield. And so, the milk fat globule, measuring a mere 3 to 6 micrometers in average diameter, is released into the glandular lumen. It is now surrounded by a phospholipid trilayer, a triple-layered marvel, rich with associated proteins, carbohydrates, and additional lipids, all derived primarily from the membrane of the secreting lactocyte. This intricate trilayer, this biological masterpiece, is collectively what we know as the milk fat globule membrane. This precise and highly specialized secretion process is a universal constant across all mammalian milk, from the human mother to the bovine. It is a fundamental mechanism of life, ensuring that the critical nutritional components of milk are delivered in their most effective form. And because this mechanism is exclusive to lactocytes, MFGM remains an exclusive treasure of milk, a testament to the specialized brilliance of mammalian biology. Where do we find this precious component? MFGM is a naturally occurring, structurally complex, bioactive component found in human milk, as well as in the milk of other mammalian species. While compositional differences do exist between species, it's fascinating to note that bovine MFGM, which has been extensively studied, shares a remarkably similar lipid and protein composition to human MFGM. This similarity is profoundly significant, as it allows us to leverage bovine sources for supplementation where human milk is not available or where additional benefits are desired. Quantitatively, MFGM makes up an estimated 2 to 6% of the total fat globules in milk. To put this into perspective, raw milk typically contains around 4% total fat, meaning MFGM constitutes approximately 0.08% to 0.24% of the raw milk itself. This seemingly small percentage belies its immense biological power. To obtain a mere kilogram of concentrated MFGM, one would need anywhere from 417 to 1250 kilograms of raw milk, a staggering amount, underscoring its concentrated potency. The content of MFGM in dairy products, however, is not static. It varies significantly depending on the processing involved. During common dairy processes like churning or decreaming, the delicate MFGM structure can be disrupted. When this happens, its valuable components are preferentially distributed into the aqueous phases of milk, such as buttermilk, butter serum, or certain types of whey. This seemingly disruptive process actually provides a serendipitous opportunity. These aqueous byproducts, rich in MFGM components, can then become excellent sources for its extraction and subsequent addition into other food products. A prime example of this application lies in infant formulas. Traditionally, infant formulas often lack the MFGM fraction because it was lost during standard dairy processing. However, recent technological advancements have revolutionized this. Scientists have developed sophisticated methods to separate MFGM from the fat globule, allowing bovine MFGM to be added back into formulas in a concentrated form. This means that today, MFGM is commercially available and can be incorporated into infant formula or indeed into a wide array of other nutritional products, enriching them with its remarkable health-promoting properties. At a structural level, the milk fat globule is surrounded by that distinctive phospholipid trilayer, teeming with associated proteins, carbohydrates, and lipids. This trilayer, as we've discussed, is the MFGM. While it may only account for a small percentage of the total milk fat globule by weight, it is an extraordinarily rich source of phospholipids, containing the vast majority of total milk phospholipids. In stark contrast, the inner core of the fat globule is predominantly composed of triacylglycerols, the simple fats we typically associate with energy storage. The MFGM structure is a masterpiece of complexity, a finely tuned orchestra of various phospholipids, glycolipids, proteins, and glycoproteins, alongside cholesterol and other lipids. What makes it even more remarkable is the specific localization of these components. Different lipids and proteins are precisely positioned within different layers of the membrane. For instance, the carbohydrate chains of glycoproteins and glycolipids are strategically directed towards the outer surface of the milk fat globule, acting like antennae, ready to interact with their environment. The lipid-to-protein weight ratio within MFGM is approximately 1:1, a balanced partnership that underscores its sophisticated functionality. However, the nutritional significance of these components extends far beyond their simple structure or macronutrient category. It is defined most profoundly by the physiological role each nutrient serves within the body. While MFGM is quantitatively a minor presence within milk, meaning it contributes very little to overall energy production, its constituents confer structural and functional benefits that are nothing short of profound. Many of these nutrients are already well known for their crucial roles within the gut, the brain, and countless other systems throughout the body. The functions of other components are still being meticulously elucidated, revealing new layers of biological wonder with each passing discovery. Delving deeper into its composition, the lipid component of MFGM is a veritable goldmine of essential molecules, particularly rich in phospholipids, glycosphingolipids, and cholesterol. Phospholipids alone make up approximately 30% of the total lipid weight of MFGM. Among these, three stand out as the most prominent: sphingomyelin, phosphatidylcholine, and phosphatidylethanolamine. Together, these three powerhouses represent up to 85% of total phospholipids within the MFGM. Their importance cannot be overstated. Phospholipids and sphingolipids play absolutely central roles in cerebral neurogenesis, the birth of new brain cells, and their subsequent migration during fetal development. They are also critical architects in promoting neuronal growth, differentiation, and the formation of synapses, the vital connections between neurons, during the crucial first year of life. Other important polar lipids within the membrane include the glycerophospholipids phosphatidylserine and phosphatidylinositol. And then there are gangliosides, a unique class of sphingolipids characterized by their sialic acid and oligosaccharide side chains. Each of these lipid classes is known to perform indispensable functional roles within the body, including the robust support of gut, immune, and central nervous system development. They're the unseen heroes, quietly building the foundations of a healthy, thriving organism. Beyond the polar lipids, the outer layer of MFGM is adorned with a diverse array of proteins, both glycosylated and non-glycosylated. Proteomic analysis, the detailed study of proteins, has unveiled at least 191 distinct known proteins in human MFGM, with comparable numbers identified in bovine milk protein concentrates. While these proteins represent only a small fraction, perhaps 1% to 2% of the total milk protein content, MFGM proteins are of immense interest due to their powerful bioactive and potentially beneficial properties. It's astonishing to consider that almost half of the identified proteins have functions related to membrane and protein trafficking or critical cell signaling pathways. Among the glycosylated proteins, we find mucins, specifically MUC1, MUC4, and MUC15, along with butyrylpholin, lactidherin, and CD36. These proteins have been suggested to significantly enhance the efficiency of triacylglyceride digestion, helping the body to extract maximum nutritional value from fats. Furthermore, lactidherin and MUC1, in conjunction with a non-glycosylated protein xanthine oxidase, have been shown or strongly suggested in preclinical studies to possess potent antimicrobial properties, acting as a crucial defense against invading pathogens. This confluence of unique lipids and proteins forms a structure with unparalleled health benefits, benefits that research continues to illuminate. MFGM contains a truly unique set of components crucial for child development, including lactoferrin, immunoglobulin G, sialic acid, cholesterol, phospholipids, sphingolipids, gangliosides, and choline. Every single one of these components plays a vital role. For instance, phospholipids and gangliosides directly influence the formation and intricate folding of the cerebral cortex, the seat of higher thought. These structural developments translate directly into enhanced neuronal development and, consequently, improved cognitive abilities. It is this understanding that drives efforts to mimic breast milk's nutritional profile in infant formulas, with MFGM supplementation becoming an essential tool in achieving this goal. It's important to acknowledge, however, that a significant proportion of studies investigating the health effects of formula supplementation with MFGM have been funded, in part or in whole, by the cow's milk and formula industry. While this doesn't invalidate the findings, it's a context worth noting as we interpret the research. One of the most profound benefits attributed to MFGM is its role in brain development and cognitive function. The MFGM lipid components, particularly sphingomyelin and gangliosides, are highly concentrated in the brain, where they are instrumental in supporting synaptogenesis, the formation of new neural connections, and myelination, the process by which nerve fibers are insulated. In the central nervous system, sphingomyelin is a key component of the myelin sheath itself. Think of myelin as the protective, fatty insulation around electrical wires. It insulates axons and ensures the efficient and rapid transmission of nerve impulses throughout the brain and body. During myelination, nerve axons are meticulously wrapped with multiple layers of cell membrane by specialized glial cells called oligodendrocytes. This process accounts for a substantial portion of brain growth during late gestation and the critical first 2 years of life, a period of explosive neurological development. Remarkably, myelination can continue actively up to 5 to 10 years of age, highlighting the prolonged importance of its building blocks. Meanwhile, gangliosides are heavily concentrated within the brain's gray matter, constituting approximately 6% to 10% of the total human brain lipid mass. They are also particularly enriched at the synaptic membrane of neurons, where they are functionally involved in neurotransmission, the chemical communication between neurons, and the very formation of synapses. Brain ganglioside accretion, the accumulation of these vital lipids, occurs at an accelerated rate during the early years of life, perfectly coinciding with the most active period of myelination, axonal outgrowth, and synaptogenesis. This synchronized development underscores their critical, intertwined roles. Alongside the remarkable growth of brain size, the total brain ganglioside concentration also increases threefold from early fetal development to 5 years of age, a clear indicator of its ongoing importance in cognitive maturation. Preclinical data provides compelling foundational evidence. Liu et al. In 2014 conducted a study on neonatal piglets, examining brain development and spatial learning and memory. They found that piglets fed a formula enriched with milk phospholipids and gangliosides, designed to mimic levels found in human milk, performed significantly better in a spatial T-maze cognitive test. These piglets made choices more rapidly and with fewer errors compared to control groups, strongly implying improved spatial learning abilities. Similarly, Vickers et al. In 2009 demonstrated that administering complex milk lipids to rats from postnatal day 10 through adulthood led to significant improvements in learning and memory tasks. Conversely, a study on complex milk lipid supplementation in pregnant mice did not show a direct effect on cognitive tasks in their offspring, suggesting that the timing and direct administration to the developing or mature organism may be key. Moving from preclinical models to human subjects, several clinical trials have aimed to assess the impact of diets supplemented with MFGM and its components, including gangliosides and sphingomyelin, on measures of cognitive development in pediatric populations. In some of these pivotal studies, MFGM supplementation to infant formula was shown to significantly narrow the gap in cognitive development observed between breastfed and formula-fed infants, a truly remarkable finding. Tanaka et al. In 2013, for instance, investigated the neurobehavioral effects of feeding a formula supplemented with sphingomyelin-enriched phospholipid to 24 very low birth weight preterm infants. In this double-blind, randomized controlled trial, the infants were fed either a control formula or a supplemented formula. The supplemented group had significantly higher percentages of sphingomyelin in total plasma phospholipids after 4, 6, and 8 weeks. Crucially, at 18 months, these infants showed improvements across multiple developmental measures, including significantly better scores on the behavior rating scale of the Bayley Scales of Infant Development 2, a higher novelty preference rate in the Fagan test, and improved sustained attention. Another significant study by Gernita et al. In 2012 assessed the cognitive effects of formula supplemented with a ganglioside-enriched, MFGM-derived complex milk lipid in term infants. Healthy infants, aged 2 to 8 weeks, were fed either a control formula or a supplemented formula designed to raise ganglioside concentration to human milk levels. A breastfed reference group was also included. The results were striking. Serum ganglioside levels in the supplemented group were significantly higher than in the control group at 6 months and did not significantly differ from levels in the breastfed group. Even more importantly, cognitive outcomes measured using the Griffith's Mental Development Scale showed that the supplemented group had significantly increased scores for hand and eye coordination, performance, and total score at 6 months compared to the control group, with no significant differences compared to the breastfed reference group. Timmy et al. In 2014 also explored the impact of MFGM supplementation on cognitive development in term infants. In their double-blind, randomized controlled trial, infants fed an MFGM supplemented formula until 6 months of age exhibited significantly higher mean cognitive scores at 12 months compared to the control group, and these scores were not significantly different from the breastfed reference group. And the cognitive benefits are not limited to infancy. Veerman-Waters et al. In 2012 assessed the potential behavioral benefits of MFGM supplementation in young children, specifically preschool-aged children between 2.5 and 6 years. After 4 months, parents of children receiving the MFGM supplemented formula reported significant differences in internal, external, and total behavioral problem scores, all in favor of the supplemented group. While teachers did not report the same magnitude of differences, the parental reports suggest a tangible positive impact on behavior and emotional regulation, offering a fascinating glimpse into MFGM's broader developmental support. This brings us to a natural pausing point, reflecting on the intricate origins and profound cognitive impact of this extraordinary biological component. But as we've already hinted, the story of MFGM is far from complete. Its influence extends well beyond the brain, reaching into realms of physical performance, immune resilience, and even cardiovascular well-being. So, let us allow these initial insights to resonate as we prepare to explore the broader, holistic influence of MFGM in our next segment. Last time, we journeyed into the very beginnings of life's most profound and intricate elixir, tracing the unique genesis of the milk fat globule membrane, or MFGM, from the specialized cells of mammals. We uncovered how this delicate, yet resilient, structure emerges, carrying with it a symphony of bioactive compounds crucial for sustenance and growth. Now, as we deepen our exploration, we turn our attention to the exquisite complexity of MFGM's molecular architecture. Imagine a microscopic blueprint, meticulously drawn by nature, where every lipid, every protein, every carbohydrate plays a vital role in constructing the very scaffolding of brilliance within us. This isn't merely about nutrition. It's about the intricate dance of molecules that orchestrate cognition, fortify our defenses, and even influence the very rhythms of our heart. At its core, MFGM is an intricate tapestry of lipids and proteins, a sophisticated boundary that encases milk fat globules. But this boundary is no passive barrier. It is a vibrant, active source of compounds, each with a profound role to play in the grand symphony of human biology. Among these are the phospholipids, sphingomyelins, and gangliosides, names that to the uninitiated might sound like abstract chemical formulas, but to the brain they are the very language of development, memory, and function. These are not merely building blocks. They are architects, sculptors of neural pathways, and guardians of cellular integrity. Think of phospholipids. These remarkable molecules form the fundamental framework of every cell membrane in your body, providing both structure and fluidity. In the context of MFGM, they are particularly abundant, especially sphingomyelin, phosphatidylcholine, and phosphatidylethanolamine, together comprising up to 85% of total phospholipids within this unique membrane. These aren't just any phospholipids. They are a concentrated source, far beyond what you might find in other dietary fats. Their presence here hints at a specialized purpose, a role finely tuned for intricate biological processes. And what a purpose it is! Phospholipids and sphingolipids, in particular, are central figures in the cerebral neurogenesis, the birth of new neurons, and migration during fetal development. Imagine, even before birth, these components are actively guiding the very formation of the brain, ensuring that neurons find their correct places, forming the complex circuits that will one day allow for thought, emotion, and action. After birth, this foundational work continues, as these same lipids promote neuronal growth, differentiation, and synaptogenesis, the creation of new connections between neurons, throughout the crucial first year of life. This is the period of explosive brain development, and MFGM components are right at the heart of it, fostering the very capacity for learning and understanding. But the story doesn't end with phospholipids. We also find the fascinating gangliosides, sphingolipids that carry an additional cargo of sialic acid and an oligosaccharide side chain. These are exceptionally concentrated within the brain's gray matter, making up a significant portion, approximately 6% to 10%, of the total human brain lipid mass. And where do they gather most densely? At the synaptic membranes of neurons, the very junctions where nerve impulses are transmitted. Here, gangliosides are functionally involved in neurotransmission and the formation of synapses, essentially facilitating the communication highways of the brain. The accretion of brain gangliosides accelerates dramatically in the early years of life, perfectly coinciding with the most active periods of myelination, axonal outgrowth, and synaptogenesis. It's a beautifully orchestrated growth spurt, where total brain ganglioside concentration can increase threefold from early fetal development to the age of 5. This is the period where the brain is literally forging its intellectual destiny, and MFGM provides crucial support for this monumental task. The scientific community has not overlooked this intricate dance of molecules. Over 2,000 scientific publications have delved into the nutritional and functional benefits of MFGM, a testament to its profound biological significance. Clinical trials, the gold standard of medical research, have further illuminated its impact. For infants, MFGM supplementation has been shown to support cognitive development, helping to bridge the gap between breastfed and formula-fed babies. In adults, it has been linked to improvements in memory and mood, suggesting a lifelong benefit. And for older adults, MFGM offers a promise of enhanced muscle strength, balance, and agility, hinting at its broader systemic influence beyond the brain. Preclinical studies, conducted in controlled laboratory settings, have provided fascinating insights into the mechanisms at play. They've demonstrated the effects of MFGM-derived bioactive components on brain structure and function, intestinal development, and immune defense. These studies serve as the foundation, allowing us to understand the precise molecular pathways through which MFGM exerts its beneficial effects. Consider, for instance, the exploration of MFGM's impact on learning and memory. In a compelling study by Liu and colleagues in 2014, neonatal piglets were fed a formula enriched with milk phospholipids and gangliosides, mirroring the levels found in human milk. These piglets, when challenged with a spatial T-maze cognitive test, made choices more rapidly and with fewer errors compared to their control counterparts. This observation strongly implied improved spatial learning, a critical component of cognitive function. Similarly, Vickers and his team, in 2009, demonstrated that providing complex milk lipids to rats from early postnatal life into adulthood led to significant improvements in their performance on various learning and memory tasks. These findings, though in animal models, offer compelling evidence for MFGM's role in shaping cognitive abilities. But the story gains even more weight when we look at clinical data, studies involving human subjects. Here, the evidence for MFGM's impact on brain development and cognitive function becomes profoundly significant. Several studies have focused on pediatric populations, exploring the effects of MFGM and its components on cognitive development. In 2013, Tanaka and colleagues conducted a double-blind, randomized controlled trial involving 24 very low birth weight preterm infants. One group received a control formula, while the other received a formula supplemented with milk-derived phospholipids, notably enriched in sphingomyelin. The results were remarkable. Infants fed the supplemented formula showed significantly higher percentages of sphingomyelin in their plasma phospholipids, indicating that the dietary supplementation was indeed being incorporated into their systems. More importantly, at 18 months of age, these infants displayed improvements across multiple developmental measures, including better scores on the behavior rating scale of the Bayley Scales of Infant Development 2, enhanced novelty preference in the Fagan test, reduced latency in visual evoke potentials, and improved sustained attention. These are not minor improvements. They speak to fundamental enhancements in neural processing and cognitive capacity. Another pivotal study by Gurnita and his team in 2012 focused on term infants, investigating the cognitive effects of a ganglioside-enriched, MFGM-derived complex milk lipid. Healthy infants were fed either a control formula or a supplemented formula, designed to elevate ganglioside concentration to levels comparable to human milk. A breastfed reference group was also included for comparison. At 6 months, the supplemented group exhibited significantly higher serum ganglioside levels, akin to those in the breastfed group. Crucially, cognitive outcomes, assessed using the Griffith's Mental Development Scale, revealed that the supplemented group had significantly increased scores for hand and eye coordination, performance, and total score compared to the control group. There were no significant differences in cognitive performance when compared to the breastfed reference group, suggesting that MFGM supplementation could indeed help formula-fed infants achieve cognitive milestones comparable to those who were breastfed. The findings continued to accumulate. Timmy and colleagues, in 2014, also assessed the impact of MFGM supplementation on cognitive development in term infants. Their double-blind, randomized controlled trial involved infants fed either a control formula or an MFGM supplemented formula, alongside a breastfed reference group. At 12 months of age, the MFGM-fed infants demonstrated significantly higher mean cognitive scores than the control group, once again showing no significant difference from the breastfed reference group. These consistent findings across multiple independent studies paint a clear picture. MFGM components are profoundly influential in shaping the developing brain. But the benefits extend beyond infancy. Veerman Waters and colleagues, in 2012, explored the potential behavioral benefits of MFGM supplementation in healthy preschool children aged 2.5 to 6 years. Children consumed either a control formula or an MFGM supplemented formula for 4 months. At the trial's conclusion, parents, using a validated questionnaire known as the Achenbach system of empirically based assessment, reported significant differences in internal, external, and total behavioral problem scores, all favoring the supplemented formula group. This suggests that MFGM's influence on brain function may also translate into improved emotional regulation and behavior, even in the preschool years. These studies underscore the remarkable potential of MFGM's intricate molecular architecture to foster not just cognitive abilities, but a holistic development of the brain. The phospholipids, sphingomyelins, and gangliosides within MFGM are not just isolated nutrients. They are integral components of a complex system that supports the very genesis of our intellectual and emotional capacities. They are the silent architects, building blocks of brilliance, meticulously constructing the complex neural networks that allow us to perceive, learn, remember, and thrive. As we conclude this segment, we are left with a profound appreciation for the sophistication embedded within a substance as seemingly simple as milk. The MFGM, with its unique origin and intricate molecular makeup, stands as a testament to nature's genius, offering a powerful blueprint for cognitive development and brain power that continues to unfold across the tapestry of our lives. Next, we will venture beyond cognition to explore MFGM's holistic influence on our immunity, our mobility, and even the vital health of our cardiovascular system. The story of MFGM is far from over, and its chapters continue to reveal surprising and profound connections across the entire human experience. We've explored the subtle origins of MFGM and how its molecular architecture quietly empowers the very essence of cognition. But the story of this remarkable substance extends far beyond the intricate pathways of the brain. MFGM, in its holistic influence, touches upon fundamental aspects of our physical well-being, our resilience against illness, and even the long-term health of our most vital organs. Imagine, for a moment, the intricate dance of life within our bodies. Every movement, every breath, every flicker of immunity, it all hinges on a delicate balance, a symphony of biological processes working in concert. And within this grand symphony, MFGM emerges as a surprising conductor, orchestrating improvements in areas you might not immediately connect with milk fat. Consider, for instance, the subtle yet profound impact on our physical capabilities, our very ability to move through the world. The milk fat globule membrane has shown a remarkable capacity to support physical performance and neuromuscular health across the entirety of our lifespan. It's not just about sharp minds, it's also about strong bodies and agile movements. The phospholipids within MFGM, particularly those sourced from bovine milk, have demonstrated tangible benefits in enhancing muscle strength, improving flexibility, sharpening balance, and refining agility. These effects, while perhaps unexpected, are deeply rooted in MFGM's fundamental role in bolstering neuromuscular junctions. Think of these junctions as the critical communication hubs between your nervous system and your muscles, the very points where electrical signals translate into physical action. As we navigate the journey of aging, a natural decline in muscle quality and neural activation often leads to reduced mobility and an increased sense of frailty. It's a gradual, often imperceptible erosion of our physical autonomy. Yet, clinical studies are beginning to reveal that MFGM supplementation can act as a countercurrent, a subtle force pushing back against these age-related declines. A randomized controlled trial, a beacon of scientific rigor, found that daily supplementation with MFGM, when combined with regular exercise, significantly improved muscle strength and overall physical performance in older adults. This wasn't merely a theoretical observation. It was a measurable improvement in the quality of their lives. Another compelling study reported enhanced agility and neuromuscular function in otherwise healthy adults, suggesting that the benefits extend beyond those facing age-related challenges. And in a more recent pilot study, MFGM supplementation, again paired with exercise, was shown to improve vertical jump power and leg strength even in young adults. This suggests a potential for optimizing athletic performance and physical vitality across different age groups. Furthermore, additional research has hinted at improvements in flexibility and balance for older adults, and even enhanced coordination in both infants and seniors when MFGM is integrated with physical activity. It speaks to a profound, foundational support for the mechanics of movement itself. But MFGM's reach extends even further into the very heart of our body's defenses, our immune system, and the intricate ecosystem of our gut. The bioactive protein components within MFGM, those sophisticated glycoproteins like lactoherin, MUC1, and butyrophyllin, have been demonstrated in preclinical studies to actively influence our immune response. They act as strategic agents, interfering with the adhesion of harmful microbes to our intestinal linings, exhibiting bactericidal properties, fostering the growth of beneficial microbiota, and subtly modulating other crucial aspects of our immune system. Consider the intestinal mucus barrier, a crucial line of defense against the constant barrage of pathogens we encounter. MFGM phospholipid components, such as phosphatidylcholine, are a vital constituent of this barrier. By supporting its integrity, MFGM may actively contribute to our intestinal defense against invasive pathogens. Then there are the sphingolipids, including sphingomyelin, which are integral to the apical membrane of our gut epithelia. They are not merely structural elements. They play a critical role in maintaining membrane structure, modulating growth factor receptors, and even acting as competitive binding inhibitors for various microorganisms, microbial toxins, and viruses. And the gangliosides, those complex sphingolipids we discussed earlier in the context of brain development, are also present in our intestinal mucosa, potentially contributing to a healthier gut microflora and bolstering our antibacterial defenses. Preclinical data paints a fascinating picture of MFGM's multifaceted immune-modulating capabilities. It can influence gut function through distinct, yet complementary mechanisms. The glycosylated proteins and sphingolipids from MFGM may actively promote the development of a healthy gut microbiota, specifically favoring beneficial bifidobacterium species. This is crucial, as a balanced gut microbiome is a cornerstone of overall health. Furthermore, a key to MFGM's immunomodulatory power lies in its structural similarity to the intestinal cell membrane. This resemblance allows human milk glycans, including those found on glycoproteins and glycolipids, to competitively inhibit the binding of pathogens, bacteria, viruses, and even toxins to our host cells. It's a remarkable example of nature's elegant design, a preemptive defense mechanism built into the very substance of milk. Numerous preclinical studies have showcased MFGM's inhibitory effects against a range of pathogens. Both whole bovine MFGM and its extracted lipid components have been shown to exhibit a dose-dependent inhibition of rotavirus infectivity in vitro. The implications are significant. Antibacterial effects of MFGM have included decreased gastric colonization and inflammation after H. pylori infection in mice, a reduction in the expression of the Shiga toxin gene by E. coli 0157, H7, and decreased colonization and translocation of L. monocytogenes. In a striking demonstration, mice that were prophylactically fed with a bovine whey glycoprotein fraction, which includes MFGM proteins, did not develop diarrhea after exposure to rotavirus. These studies provide compelling evidence of MFGM's proactive role in protecting against infectious agents. The clinical data further solidifies these findings, demonstrating tangible benefits in human populations. A study examining the effects of MFGM supplementation in term infants revealed a significant reduction in episodes of acute otitis media, a common ear infection, in the MFGM supplemented group compared to those fed a control formula. This reduction was so substantial that the incidence mirrored that of breastfed infants. Moreover, a significantly lower incidence and longitudinal prevalence of antipyretic drug use, medications for fever, was observed in the MFGM supplemented group. This suggests not just a reduction in specific infections, but an overall strengthening of the immune response. Further research linked this benefit to changes in the infant's oral microbiota, with Moraxella catarrhalis, a common bacterial culprit in otitis media, being less prevalent in the MFGM supplemented infants. Another critical study, conducted in Peru, evaluated the effects of an MFGM-enriched complementary food on health outcomes in breastfed infants. The results were clear. The MFGM supplemented group experienced a significantly lower prevalence of diarrhea during the study, as well as a significant reduction in episodes of bloody diarrhea. Subsequent analysis of these infants' metabolome and immune markers revealed that MFGM supplementation might improve micronutrient status, amino acid, and energy metabolism, alongside a reduced pro-inflammatory response. In extending to preschool-age children, MFGM supplemented formula consumption led to a significant reduction in the number of days with fever, particularly short febrile episodes. These collective findings paint a powerful picture of MFGM as a robust protector of infant and child health, bolstering immunity and promoting a healthier gut environment. Finally, we arrive at an often debated frontier of health: cardiovascular well-being. For decades, dietary guidelines have often advised limiting full-fat dairy products, largely based on the traditional hypothesis that dietary saturated fatty acids, abundant in milk fat, elevate serum LDL cholesterol, a known risk factor for cardiovascular disease. However, a deeper understanding of dairy components, particularly MFGM, is beginning to challenge and refine this perspective. Observational studies now suggest that the association between milk fat intake and serum cholesterol measures can vary significantly depending on the type of dairy products consumed. This variation might, in part, be attributed to the nuanced presence and composition of MFGM. The lipid components of MFGM may play a crucial role in supporting cardiovascular health by modulating cholesterol and fat uptake within the body. Preclinical data provides compelling evidence for this. MFGM lipid components like sphingolipids are directly involved in the intestinal uptake of cholesterol. Studies in adult rodents have shown that milk sphingomyelin can lower the intestinal absorption of cholesterol in a dose-dependent manner. In other words, the more sphingomyelin, the less cholesterol absorbed. This mechanism involves sphingomyelin and other phospholipids with a high affinity for cholesterol, which can limit the micellar solubility of intestinal cholesterol, thereby restricting its uptake by the enterocyte cells lining the gut. Furthermore, dietary sphingolipids have been shown to dose-dependently lower plasma cholesterol and triacylglycerol levels in adult rodents fed a Western-type diet, and even protect the liver from fat and cholesterol-induced steatosis or fatty liver. These dietary sphingolipids also reduce hepatic cholesterol and triglyceride levels in adult rodents, partly by modulating hepatic gene expression. The clinical data, while still emerging, offers intriguing insights into MFGM's potential cardiovascular benefits in humans. A single-blind, randomized, controlled trial in overweight adults showed that the effects of milk fat on plasma lipids were notably modulated by the MFGM content. Strikingly, compared to butter oil, which served as a control diet, the consumption of whipping cream, an MFGM-rich diet, for 8 weeks did not impair the lipoprotein profile. This suggests that the presence of MFGM can mitigate some of the traditionally perceived negative impacts of saturated fats. Another double-blind, randomized, controlled trial in overweight and obese adults further demonstrated that MFGM attenuated the negative effects of a high-saturated fat meal by reducing postprandial cholesterol, inflammatory markers, and insulin response. This speaks to a protective effect, tempering the acute physiological stress caused by such a meal. And a third double-blind, randomized, controlled trial in normal healthy adults indicated that just 1 month of consuming buttermilk, naturally rich in MFGM, led to a reduction in both serum cholesterol and triglycerol levels, as well as blood pressure. Perhaps most profoundly, there's a hypothesis that MFGM supplementation in infancy may have profound programming effects that could influence circulating lipid levels later in life. We know that breastfed infants tend to have higher total serum cholesterol and LDL cholesterol than formula-fed infants during their early months, yet paradoxically, they exhibit lower levels in adulthood. A clinical study in infants suggested that MFGM supplementation could narrow this gap between breastfed and formula-fed infants concerning serum lipid status. Specifically, infants receiving MFGM supplemented formula had higher total serum cholesterol up to 6 months of age, mirroring the levels seen in breastfed infants, compared to a control formula. While the LDL-HDL ratio didn't differ between the formula-fed groups and was higher in the breastfed reference group, this subtle alignment in cholesterol trajectories in early life points to a deeper, long-term influence on metabolic programming, underscoring MFGM's potential not just for immediate health, but for shaping future well-being. As we bring this exploration of the milk fat globule membrane to a close, it becomes clear that MFGM is far more than a mere structural component of milk. It is a biological marvel, a complex tapestry of lipids and proteins woven with profound purpose. From orchestrating the intricate dance of brain development and cognitive function, to bolstering our immune defenses and the delicate balance of our gut, and even extending its protective embrace to our cardiovascular system, MFGM emerges as a truly holistic contributor to health. Its story is a testament to the elegant ingenuity of nature, reminding us that even in the most seemingly ordinary substances, extraordinary science can be found, waiting to be understood, to be appreciated, and to quietly enhance the quality of our lives. And so, our journey through the microscopic universe of the milk fat globule membrane draws to a close. From its genesis within specialized cells, to its remarkable role in sculpting our brains, fortifying our defenses, and even guarding our hearts, MFGM stands as a testament to nature's profound ingenuity. It reminds us that even in the most familiar corners of our world, hidden wonders await discovery, continuously reshaping our understanding of life itself. I hope this deep dive brought you both knowledge and tranquility. What are your thoughts on this incredible component of milk? And what other scientific mysteries would you like us to explore next? Please share your insights in the comments below. Like this video if you enjoyed the exploration, and follow for more journeys into the cosmos, the quantum, and everything in between. Until our next scientific sojourn, keep exploring, keep questioning, and keep vibing with science.